Assalamu alaikum, everyone. In the previous lecture, we started learning about the transposable elements and their related sequences. As you saw in the previous lecture, almost 55 to 59% of our genome consists of repetitive sequences. So if I go back to this pie chart here, these repetitive sequences are divided into two major types. We have the repetitive DNA sequences that consist of transposable elements, and the repetitive DNA sequences that are not related to transposable elements. And we discussed transposable elements in the previous lecture. Today, inshallah, we'll continue discussing the different types of repetitive DNA elements. So 5 to 6% of these repetitive elements, they consist of large segments that are duplicated many times. Okay. So we discussed transposable elements and how they the mechanism by which there are more copies of these transposable elements are generated, whether they are transposons or retrotransposons. And still, we have some large DNA segments are duplicated, and these DNA segments, they represent 5 to 6% of our genomes. Now, these long sequences, as mentioned in your book, their size ranges between 10,000 to 300,000 base pairs. Okay, so they could be duplicated. Duplicated means مضاعفة, and they could be duplicated and another copy is inserted on the same chromosome or a different chromosome. Okay. And these segments, these long sequences, they might contain genes. Okay. So it's not necessarily non-coding DNA. They might contain some genes, which means that some genes might be also duplicated, as we will see, inshallah, today, how they are going to be duplicated. Some genes are actually duplicated within our genome. Okay. The other sequences that are found, the other types of repetitive sequences that are unrelated to transposable elements are symbol sequences, which represent 3% of our genome. Okay. These symbol sequences are simply short sequences that are repeated hundreds or maybe thousands of times. And they are repeated in tandem. Okay, so for example, in your book, you have this sequence like GTTA C. This could be repeated hundreds or thousands of times. Okay, many times. Okay. So as you can see, we have, this is the repeating unit. The repeating unit, this is basically the sequence that is repeated in tandem. In tandem means one following the other. Okay. So here we might have in between n number of repeating units. Okay, the rest of the genome, the rest of the genome. So this is the repeating unit consisting of 5 nucleotides. It could be repeated hundreds or thousands of times. And this repetitive DNA could be found more than once in different locations within the genome. Okay. Some of these simple sequences, they might contain from 2 to 5 nucleotides. And they are, in this case, they are called short tandem repeat, or STR. So short tandem repeats, they are scattered. They consist of simple sequences consisting between 2 to 5, repeated hundreds or thousands of times within the same location, and these repeats are found scattered throughout the genome. Okay. These STRs actually, they are important for forensic science. العلوم الشرعية, because they could be used forensic science, because they could be used to detect, because the location of these short tandem repeats within the genome is different in different people, and the number of these the number of times these short tandem repeats are repeated within a specific location within genome will be different between humans. Okay, that's why forensic or geneticists or forensic scientists, they rely on the STR analysis to determine, for example, if a sample from a crime scene, let's say من جريمة قتل, من مسرح جريمة, could be matched with a specific suspect. Okay. أو إذا حدا ارتكب جريمة ممكن يستخدموا ال STR analysis حتى يعرفوا مين اللي ارتكب الجريمة. Remember also we have these simple sequences also include the telomere sequence. Remember the telomere sequence. We know that at the ends of chromosomes, we have repeated sequences called telomeres. And in humans, the telomeric sequence is basically consists of 6 nucleotides. So this is the repeating unit this repeating unit is repeated hundreds of times, maybe thousands of times in both ends. So approximately it is repeated up to 3,000 times in both ends. Okay. So these simple sequences, these repeats are they are common in centromeres. Okay, so this is the centromere region. And they are important, they have structural roles. So we know that the centromere is important for the binding of certain proteins like the mitotic spindle or the spindle fibers of during cell division or during mitosis and meiosis. And these telomeres, they maintain the structure of the chromosome and they prevent the damage of genes, because chromosomes, they become shorter and shorter after each cell division. Okay. The telomeres are, yes, they are found at both ends of the chromosome, but other simple DNA sequences or other types of repeats might be found also on the centromere. And short tandem repeats, they might be found, let's say, at different locations within, let's say, the chromosome. So this is STR, this is STR, and STR. And these STRs, they are also found in other chromosomes. So all of these, the repetitive DNA, the simple repetitive DNA in the centromere, in the telomeres, in the STRs, they are all part of the 3% of simple repetitive sequence DNA that is not related to transposons. We have also what we call multi-gene families. Here, some genes, some eukaryotic genes, they might be present in our genome in more than one copy. So most of our genes are present in one copy per haploid set. So the idea here is that from most of our genes, we have 2 copies. One copy you inherit from your father, the other copy you inherit from your mother. But for some other genes, we might have more than one copy found in the genome. So more than one copy per the haploid set of chromosomes. So the haploid set here, what is meant by the haploid set, this is basically let's say the chromosomes inherited from one parent. يعني هالمجموعة the haploid are the chromosomes that you inherit from either your father or your mother. This is what is meant by one copy per haploid. Okay. But some genes, there might be present in more than one copy per the haploid genome. So for example, in eukaryotic genomes, most of our genes are solitary genes. They are called solitary genes. So these are one copy per haploid set. Okay. While some genes, they are part of what we call multi-gene family. So we have here we're talking about 2 or more. Yes, here we have 2 or more identical or very similar genes. So they represent, that's why they are called gene family, because their sequences are either identical, mutatabiq, or very similar. Okay, solitary genes, here we're talking about the genes you inherit one copy per haploid set. So basically you have 2 copies of these genes, one copy from your mother, one copy from your father. Multi-gene, you might here inherit multiple copies from your father and multiple copies from your mother. Now, again, these genes that could be identical, they could be identical DNA sequences. And usually these genes, the multi-gene families that consist of identical sequences, they cluster tandemly. Tandemly means they are found, cluster means بيتجمعوا. Tandemly means they are found within the genome cluster, grouped together, one following one. Okay. An example of clusterly clusterly and tandemly, let's say, multi-gene families is the gene family of the rRNA genes. So here we have, for example, the genes encoding the rRNA, the ribosomal RNAs, which basically these genes, or the rRNA gene, they are transcribed, but they are not translated. So you have hundreds of copies of these genes clustered tandemly or repeated tandemly. You have hundreds or thousands of them. So this is, for example, one rRNA gene. This is another. This is another. And there will be hundreds. And all of them are repeated in the same region and in tandem. Tandem means one following one. So this is the transcription unit. This is the rRNA. This is the rRNA gene. This is another rRNA gene. And as you can see, this is an electron microscopy image showing active transcription. And between these genes, we have non-coding DNA. This is non-transcribed spacer. So this is DNA we're talking about here. This is the microscopic image of the rRNA genes, DNA. And here we can see active, very active transcription, because we see the transcripts are being generated. And these transcripts are then divided into 3 rRNA molecules, like this. And these 3 RNA molecules are called 18S, 5.8, 25S. They are basically important components of the ribosome. So this is the ribosome. That's why they're called ribosome. That's why they are called rRNA. R stands for ribosomal R. Okay. Some other gene families, they consist of genes that are not identical. They are very similar genes. So here, the sequence of this gene, this gene, this gene, this gene, and this gene, and maybe other 100 genes, they are identical in sequence. Okay. But for other multi-gene families of other multi-gene families, they might have non-identical gene sequences. An example of multi-gene families with non-identical genes or very similar gene sequences are the alpha globin multi-gene family and the beta globin multi-gene. These are the proteins or the polypeptides that are important for the formation of hemoglobin. Remember, the human hemoglobin consists of 2 alpha and 2 beta polypeptides. So this is hemoglobin. And we have the beta globin gene that will give the beta polypeptide. And we have the alpha globin gene that will give the alpha polypeptide, 2 beta polypeptides, okay, like here and here, and 2 alpha polypeptides. They come together to form the human hemoglobin. we have two multi-gene families, alpha globins and beta globin. So the alpha globin consists of a number of genes. Some of these genes are pseudogenes. So these are the green, the yellow genes are actually pseudogenes. Remember, pseudogenes are, so these are pseudogenes. Just to remind you, pseudogenes, they used to be genes, but they accumulated many mutations, so now they are non-functional. So these are non-functional genes. Non-functional gene. Okay. And we have, for example, the alpha globin gene family located in chromosome 16. Okay. And they consist of a number of genes. As you can see, we have 7 genes. 4 of these genes are pseudogenes, but also they have other genes, like we have alpha 1 and alpha 2. Okay. And we have also zeta genes. Okay. So this zeta gene is found, encodes a globin protein that is found in the hemoglobin in the embryo. The alpha and the alpha 2 genes encodes polypeptides that are found in the adult hemoglobin. So the hemoglobin in adults, بالبالغين, is different than the hemoglobin, is made of different polypeptides than the ones that is found in the embryo, بالجنين. Okay. Beta globin gene family found in chromosome 11, and it contains, for example, epsilon gene, which is basically encoding a polypeptide that is part of the embryo hemoglobin. We have also 2 genes, 2 gamma genes that are found in the fetus embryo, in the fetus hemoglobin. And we have the beta, which is beta gene, which encodes the beta polypeptide that is found in the adult hemoglobin. And we have also the sigma gene encoding sigma globin, which is also found in some hemoglobin proteins in India. So as you can see here, we have 7 genes. We have 7 genes, different chromosomal location. This is one family. So the nucleotide sequences of all of these genes, so each one is considered a gene, and the gene sequences, or the sequences of all these genes, they are not identical, they are very similar. Okay, it's very important. Not identical, but very similar gene sequences. And there are many other example gene families. Gene family or gene families found in the human genome or the genomes of families. All right. So in the next topic, we're gonna see, or we're going to learn about some mechanisms that might lead to the change in the structure of the genome, to the evolution of the genome. So mutations are actually the major cause of genome evolution. But we have also other mechanisms. So mutations, they will either, for example, make some genes pseudogenes, or they might also generate new genes with the new encoding proteins with the new functions. Okay. So the first mechanism we're going to discuss is the duplication of entire chromosomes or whole chromosomes. So sometimes errors during meiosis can lead to one or more extra sets of chromosomes finding their way into one of the cells that are produced during meiosis. So remember what you've learned during meiosis. Meiosis is used to basically to produce cells that have the let's say half the number of chromosomes of the mother cell. So during meiosis, you have a cell that is diploid, okay, undergoes, let's say, meiosis 1, okay, 2 diploid cells are generated after meiosis 1. Okay. And then what happens is that after meiosis 2, you have 4 cells generated. Each one of them is haploid. So 1N means haploid, 2N means diploid. So this is 2N. These are haploid cells. In humans, if we start with a diploid cell containing 46 chromosomes, during meiosis or after meiosis, we have haploid cells, each containing 23 chromosomes. Of course, this diploid cell, before it undergoes meiosis, it should undergo a process called DNA synthesis. Okay, so to double the amount of DNA found in the cell, but still it's 2N. So here, before meiosis, each chromosome is duplicated. Okay, and each chromosome consists of 2 sister chromatids. So at the end of meiosis, we have half the number of chromosomes. Okay. Now, polyploidy happen when errors happen during meiosis, And these errors could lead to the generation of gametes having more than one set of chromosomes. So you can imagine that here, these cells must be haploid, and these are called mainly gametes. Okay. And some of these haploid cells might contain, so errors might happen during meiosis, and these errors might result in the generation of not one N, let's say, chromosomes, but two N, sorry, not one N gametes, but two N gametes. Okay, so errors might happen here, or might happen here in meiosis 1 or in meiosis 2. Okay. So if, for example, if this cell, if the chromosomes in this cell do not segregate, we will end up with two cells. Come on. Two cells might have, one of them will have 2N, let's say, and one nothing. And imagine if this haploid gamete, if this is maybe an egg, maybe this is female gametes, let's say they are eggs, and this egg is fertilized with a sperm, okay? The result would be a zygote that is 3N. So having a 2N, let's say, if we're talking about human chromosomes, means it will have 46 chromosomes, and this is a haploid sperm, okay? So maybe this is an egg, and this is a sperm, and the sperm will contain 23, and now we will end up with a zygote that contains 69 chromosomes, and 69 chromosomes, they represent 3. Of course, the zygote will die, it will not develop into a whole organism. But some organisms, they can tolerate having more than one chromosomal sets, especially plants, okay? So this is called polyploidy, having more than one extra set. So the zygote should contain only 2 sets, but now it contains 3 sets of chromosomes. This is called polyploidy, okay? Plants, they can tolerate polyploidy. So plants, they can have more than 2 sets of chromosomes, more than animals. Actually, many of the different fruits or vegetables you eat, actually they are polyploid, let's say, organisms. So, for example, strawberry, they might contain, let's say, some types of strawberries, they are 10N, 10 sets of chromosomes, okay? Some types of, for example, kiwi contains 6N, or 6 sets of chromosomes. So there are like, so this is called 10 ends means we have decaploid. Deca means 10. 6N means hexaploid. Some types of wheat, qamh, they are hexaploid. Some types of kiwi, hexaploid. Some types of strawberry, they are decaploid, 10 ends, or octaploid, which means 8N. Okay. Yes, in plants, in nabatat, many plants, especially the plants that we're using as a food source, they are polyploid, actually. And actually, for example, if you see large strawberries, strawberry ضخمة, these, like in general, as a general rule, of course, there are some exceptions. Polyploidy increases the size of fruits or vegetables, typically. Okay? But in animals, polyploid zygotes, they do not develop into organisms. They die at an early stage of embryogenesis or development. Now, here we're talking about So here we're talking about duplication of a whole set of chromosomes, adding extra set or more than one set. But also duplication might happen to one chromosome or parts of the chromosome. So when scientists analyzed the human chromosome number 2 and 2 chromosomes from chimpanzees, which are types of monkeys, let's say, they found out that human chromosome number 2, okay, is similar. So here we have the chromosome telomere sequences at both ends. We have a centromere, and there is an extra region here that is similar to the telomere, and there is an extra region here that's similar to the centromere. So scientists, they think that we are different from the chimpanzees. In the chimpanzee, the number of chromosomes in humans so we know that the number of chromosomes in humans is 46. Okay. In chimpanzees, they have 48 chromosomes. Okay, we have 46. The difference is that they have these two. So chromosome number 12 is very similar to this part of chromosome number 2, and chromosome number 13 is very similar to this part of chromosome number 2 in humans. Okay, so scientists, they think that in humans, in chimpanzees, these two are separate chromosomes, they remain separated, but in humans, these two chromosomes, they represent one chromosome. Okay. So scientists, they hypothesize that these two chromosomes in humans are fused together. That's why we have 46 chromosomes, and chimpanzees, they still have these two chromosomes separate. That's why they have a higher number of chromosomes. Okay. Also, when scientists analyzed some mouse chromosomes, chromosome 7, 8, 16, 17. They found different regions in these 4 chromosomes that corresponds to regions in chromosome 16. Okay. So here we can see that we have similarities in terms of the DNA segments within chromosomes from humans and chromosomes from other organisms. Now, we could also have some duplications and inversions. Inversions means the orientation of the segment of DNA changes. It changes because of mistakes during mitotic recombination. Remember that when you learn about meiosis, in meiosis 1, during the prophase 1, we have something called crossing over. Remember that? Okay. This is called meiotic recombination. And meiotic recombination or mistakes in meiotic recombination might lead to duplication. How? These are, so for example, these are 2, okay, let me call them like this. These are 2 homologous, or let's call them duplicated homologous chromosomes. What do I mean by duplicated homologous chromosomes? Do you remember? Do you remember from Biology 101 what is meant by duplicated homologous chromosomes? Anyone? So these homologous chromosomes, they are coming from different parents. They are called homologous chromosomes because they have the same length, they are carrying the same types of genes, and they have the same banding patterns when they are synced. Okay? Not similar alleles, similar types of genes. Okay? That's why they're called homologous chromosomes. But why they're called duplicated? Why they are duplicated? Why they should be duplicated during meiosis? So each chromosome consists of what? Here we have these 2 are called sister chromatids. Right? Because remember, in meiosis, we start with 46 chromosomes, 1 cell. After meiosis 1 and 2, we will end up with 4 cells, each one 23 chromosomes. So chromosomes, here we have, let's say, a chromosome, and here we will have duplicated chromosome after DNA synthesis, after the S phase of the interphase stage. Okay, so here we have 2 sister chromatids, and then these sister chromatids will be separated during meiosis 2, for example. Now, these 2 are called non-sister chromatids. Okay. So these two homologous chromosomes, so let's say that this is, just for example, this could be, let's say, chromosome 1 from the father, and this is chromosome 1 from the mother. Now, crossing over happens during prophase 1 of meiosis 1, and crossing over happens between non-sister chromatids. So both non-sister chromatids will be aligned along their lengths like this. And then what will happen is that a crossing over might happen here. Let's say this is crossing over. And this might lead to exchange of DNA fragments. So let me use two different colors. So now we have, so we will have a chromosome like this. With another sister chromatid chromosome. And maybe something like this. So I gave the two chromosomes different colors to indicate that an exchange of DNA segments happened. Okay? So this crossing over will result in this exchange of DNA fragments. Okay, and maybe here we have a gene, some sort of a gene. Okay, so this is the crossing over happens here and will result in exchange. This region of DNA will be exchanged with this region of DNA, like this. But what happens is that, notice here, we have transposable elements, which means this is a repetitive DNA, this is repetitive DNA, this is repetitive DNA, this is repetitive DNA, okay? And these repetitive DNA, sorry, the exchange is happening here, actually. And this crossing over happening here because this DNA is identical in sequence of this DNA, which means these identical sequences could be the site of crossing over. So this is crossing over, or it is also called meiotic recombination. Meiotic recombination happens when there is identical sequences like the transposable elements. They are aligned across each other when the two non-sister chromatids align along their length. Okay? So crossing over could happen between these two in this region, or maybe in this region, because here we have identical sequences. That's why they are given the same color. But crossing over, in theory, could also happen between this sequence and this sequence, because these two sequences are also identical. And this happens when these two transposable elements are misaligned. So here the crossing over happened because, for example, because here we have correct alignment. Correct alignment means they are found in front of each other like this. So this is correct alignment. Because as you can see, both non-sister chromatids, they are aligned along their length. This is called misalignment. So this sequence, which is identical to this sequence, to this sequence, to this sequence, these two sequences this one and this one, they will come closer to each other. Okay, and now crossing over happens between these two, like this. Okay, and these are now considered misaligned chromatids. As you can see, the homologous chromosomes, they are not aligned with each other according to their length, like here. So this is correct alignment. This is incorrect pairing or alignment of the homologous chromosomes, which leads to incorrect alignment or misalignment, which might lead to crossover, because these two, or recombination, because these two sequences are identical. So recombination happens between identical sequences or very similar sequences, and that will result in this exchange of genetic material. So this part from here to here will be added to the blue chromosome, and this part from here to here will be added to the purple chromosome, like this. Now, in terms of genes, what will happen is that this chromosome, the purple chromosome, receives another copy of the gene. And the other chromosome on the other the other chromatid on the other homologous chromosome is now does not contain this gene. So this is a chromatid with deleted gene. This is a chromatid with duplicated gene. And eventually, these 4 chromatids will end up in 4 different cells or gametes. So this one will have the full, let's say these are gametes. So if my meiosis continues, this chromatid will eventually found its way into one of the gametes. And this chromatid also will go to one of the gametes. And here we have also another gamete, and then we have. So this is a parental chromosome. It's a chromosome like the parent, similar to the chromosome came from the parent. This is a parental chromosome. These are recombinant chromosomes. They contain DNA from different chromosomes, from the mother and the father. Okay. Imagine if this gamete is used in fertilization. This gamete is carrying 2 copies of the same gene. This one carrying 1 copy. This one is carrying 1 copy, and this one does not carry any copy of the gene. Okay. So if this is the gamete that is used during fertilization, this results in a zygote that having, so this is, for example, if this one is used for fertilization by a sperm, imagine if these are eggs, let's say, and this one is fertilized by a sperm, and this sperm is carrying the same chromosome. Okay, let me give it a different color just to indicate it's a similar chromosome. It's the same chromosome, but this chromosome will be carrying 1 copy of this gene. So now 1 copy of this gene is coming from the sperm, and 2 copies of this gene are coming from the eggs. So now we have 3 copies in total. So this is what is meant by gene duplication or chromosomal region duplication. So the whole chromosome, there might be other genes. So this region is there are 2 copies of this region in the same chromosome. Okay. So duplications and inversion. Sometimes you have one region. Let's say that we have a chromosome, and then we have another chromosome, sometimes what will happen is that these two chromosomes, they will exchange some segments like this. Okay, so this region of the chromosome jumps to this region, and this region of the chromosome jumps to the other region. So this is called translocation. Okay, but also sometimes what will happen is that within the same chromosome, let's say that here we have gene A and gene B and gene C and gene D. What will happen is something called inversion. Inversion is basically we will have a change in the orientation of the chromosome or DNA segments within the same chromosome. So now we have A, B, C, D. So it's like this part of the chromosome is flipped or changed its direction. Okay, these are called inversions. Of course, we discussed duplication, which is basically making more copies of genes or DNA fragments or DNA segments. So as you can see, transposable elements, they might provide sites for they provide sites for crossing over. But if the crossing over happens between the misaligned transposable elements, this might lead to chromatids with duplication and chromatids with some regions that are deleted. Now, scientists, they have a hypothesis regarding how gene families or how genes they might form a gene family or a multi-gene family. So for example, scientists, they studied the globin gene family, and they think that the globin gene family began or originated from a single gene. This is the ancestral globin gene. Ancestral means in Arabic, I don't know what's ancestral means الجد الأول, maybe we could say. Trying to find a word. Ancestral السلف الأول. Okay, and this gene was duplicated. Okay, and you have to understand that it could be duplicated because of errors during, let's say, like here during meiosis, and the duplicated chromatid was used for fertilization to generate, let's say, a new organism. Or it could be duplicated because of transposable elements. So then now we have two copies and some mutations. So these are identical here. So gene duplication, then identical copies. And then these two identical copies. So let's say this is the X gene, this is X gene, now this is X gene. And now these two copies, they have mutations, and now they have different sequences. That's why they're called alpha and beta. Then transposons might change the location of these two genes within the chromosome. So now this A gene was carried by a transposon into chromosome 16, and maybe the beta gene was carried out into chromosome number 11. And then further duplications and mutations generated all the genes within each one of these each one of these two gene families. So as you can see here, we have three mechanisms. We have duplication, we have gene mutations. So gene duplications makes another copy of the gene. Gene mutations makes a new gene, or basically it will make a new gene or change the sequence of the gene. Then we have gene transposition. So gene transposition means the gene changes its location within the genome with the help of transposon, by transposon. So transposons might carry genes into different locations within the genome, or might change their position mechanically. Okay? And these three mechanisms, scientists think they are responsible for the generation of these alpha and beta global gene family. And scientists think that all genes within these two families originated from a single ancestral gene. Okay. Gene duplications followed by mutations might generate new genes with novel functions. Novel means جديدة. Okay. So for example, scientists think that two genes, one encoding the lysozyme, which is an enzyme, and the other codes another protein called alpha-lactalbumin. They think that both genes originated from one gene. So scientists, they looked at the amino acid sequence and the structure of the lysozyme and alpha-lactalbumin, and they found that there are some similarities in terms of structure and in terms of the amino acid sequence, but there are some differences. So scientists think that the lysozyme gene, which encodes an enzyme, was duplicated and accumulated in mutations. So you could think of, here we have a lysozyme gene, was duplicated. So now we have two lysozymes. So here we have duplication. Two lysozyme genes, and then mutations affected one of the genes, which resulted in basically a new gene with a completely different function, or a new function. So this remained the lysozyme gene, and this became the alpha-lactalbumin gene. Lactalbumin gene. Albumin gene. Okay. So this gene encodes a protein that has a completely different function than the protein encoded by the lysozyme. The lysozyme gene encodes an enzyme called lysozyme. And this lysozyme can, it's an enzyme that can protect animals against bacterial infection because it can degrade the cell wall of bacteria. Alpha-lactalbumin gene, it's a protein, but it's not an enzyme. Okay, and it is found in the milk of mammals, الثدييات. حليب الثديين. Okay. So another mechanism that might lead to a change in the genome and gene structures are two mechanisms called exon duplication and exon shuffling. So here in this example, so we discussed basically polyploidy, which basically duplication of a whole set of chromosomes. Then we discussed duplication of, so we discussed duplication, inversion, translocation of parts of the chromosomes. Then we discussed duplications and deletions of genes. And now, and here also duplication of genes and translocation of genes and change in gene sequences by mutations. And now we're gonna see how exons of genes could be also duplicated and shuffled to generate new genes. And this exon duplication and shuffling could also happen by the same mechanism we explained here that resulted in gene duplication. So here we're talking about the same mechanism, errors during meiotic recombination might lead to exon duplication and exon shuffling or mixing and matching of exons. Okay, this is what is meant by shuffling, mixing and matching. Shuffling خربط. So for example, scientists found out that a gene called the TPA gene, tissue plasminogen activator gene, consists of 4 exons, F, EGF, and K exons. And scientists, they think that these exons or this gene was the result of mixing 3 different types of exons from 3 different genes. So here we have the epidermal growth factor gene consisting of 4 copies of this exon. We have the fibronectin gene with multiple finger exons, and then the plasminogen gene with the Kringle exon. And scientists think that the TPA gene was actually generated from taking and mixing different exons from different genes. So one exon came from the EGFP gene, the EGF gene. One exon came from the fibronectin gene, and an exon came from the plasminogen gene, and this exon was also duplicated. Okay? So here we have something called exon shuffling and exon duplication. Now, transposable elements might be responsible for, as we see, they are in general responsible for recombination or crossing over. So also they might be responsible for incorrect crossing over, leading to gene duplication or leading to exon shuffling and exon duplication. Okay. So I'm gonna stop here, but inshallah tomorrow we will learn how exons might play a role in let's say chromosomal segment duplication or gene duplication or exon duplication and exon shuffling. And we will see how also transposon are responsible for might be responsible for the change in also gene expression and how they affect gene expression. Okay. So let me stop here and we'll continue inshallah